Thank you so much for having me. And first, actually, let me apologize for not being there in person. I had been planning on coming there and was very, very much looking forward to it. But at some family obligations intervene at the last minute. Um, but I am really pleased to be able to share an update on some of our work with all of you. Let me go ahead and share my screen so I can get started. As we get going, I'm a neurologist and a geneticist at Boston Children's Hospital. And the work that I'm going to be sharing with you is the result of a very large collaborative effort from many folks in the AT community many of whom are right there at the conference. So I'm pleased to share it and to get your feedback. Here we go. Is my first slide showing?
Yeah.
Perfect.
Yes,
Okay.
it is.
So I'm going to be telling you about some pilot work that we've been working hard on for the last 7 years to develop proof of concept precision genetic treatments for ataxia-telangiectasia. And two key members of our team who are at the conference are Claudio and Val, who I encourage you to hunt down and pin down and chat with questions that you've got. And I also encourage people to reach out to me with any questions that you might have by email, even since I can't be there in person. My research group at Boston Children's Hospital has focused on trying to understand pediatric neurologic disease, to find better ways of discovering genes that cause these diseases, to accelerate the diagnosis of children with these conditions, to try to understand from their genes why things go wrong, and to try to develop treatments. And to frame the work that we've been doing with AT, I'd like to present one of the central challenges in this space, which is that there's a very long tail of genetic diseases like AT that are incredibly important, yet in terms of numbers are relatively rare in the world. And the sad truth is that 85% of these conditions are too rare to be considered commercially viable for the usual types of drug trials that we typically like to try to power up. And this is a real challenge in the field, and it requires us to try to think of some creative solutions. The reason for this isn't because companies don't care. The reason for this is that historically, drug making is incredibly slow and expensive. These are some statistics, granted, they are now over 10 years old, describing the incredible costs that the industry has historically borne to bring new drugs to approval. And the time it takes to go in the top right, the cost of $2.6 billion. That's a statistic from the NIH about the industry cost per drug approval, and incredible amounts of years required to do that, and a high failure rate. I don't say this to depress you, but I say this to set the baseline for why the field has been so slow to turn around. And the good news is that this is beginning to significantly change. One of the reasons for that is that there are new waves of genetically targeted treatments that are beginning to get traction. And some of those are listed here. We're talking about things like gene therapy, mRNA therapy. You heard about that during the COVID pandemic, antisense oligonucleotides. Some of these genetically targeted treatments are shown on this slide. And the remarkable thing about some of these genetically targeted treatments is that they can be developed very quickly. On the other hand, one of their weaknesses is that you can make them very fast, but you can't give them to everybody. Many of them are so precise that you can only give them to certain people with certain genetic profiles. So it is an unusual situation where instead of making one drug for everybody, there are opportunities now to make very precise drugs very quickly, but you have to make many of them. And that's one of the themes that you'll see in this approach and this pilot study that we've been doing. So as I mentioned, this genetically targeted treatment category that I'm going to be focusing on is this long word in the middle, antisense oligonucleotides. What are those? Well, first let's abbreviate it because it's too much of a headache to say. Let's call them ASOs. That's the industry parlance for them. And what are these? These are short snippets of synthetic genomic code. You can think of them as little tiny fragments of the human genome that have actually been synthesized in the laboratory and made to match different regions in our genes. They're very easy to manufacture, they're inexpensive to manufacture, and they home in on certain spots in our genome based on what their letter sequence is. As you know, the genome is based off letters A, C, G, T, and these pair up in certain ways. And depending on how you arrange the sequences of these very, very short drugs, they will home into one particular spot in the in the genome. They've been chemically modified, so they're not our natural DNA and RNA, but these modifications have helped them be good drugs. When you inject them into the body, they're well absorbed, they're stable for a long time, they're relatively safe, and you can use them in various ways. In particular, you can use them to boost gene expression by changing the way that genes are assembled or spliced. And I'll expand a little bit more on that. Here's an example of two antisense oligonucleotide drugs. The sequence in green is a series of letters, 18 letters long, that is a very famous ASO drug, the first ASO drug to really hit the market. It's a drug for a condition called spinal muscular atrophy, and it targets a gene called SMN1. The drug in blue, the ASO in blue, is a same chemistry, the same type of ASO, but a different pattern of letters. And that is a drug that we made in 2018 to target a specific mutation in a specific child that allowed us to treat her with this experimental ASO. Just a little bit more on this background before I switch to AT. As I mentioned, this is that drug for spinal muscular atrophy, that first in class ASO. And it, again, is an 18-letter ASO. It's manufactured and then it's as a powder. It's dissolved essentially in salt water or artificial cerebrospinal fluid. That's the fluid that bathes our brain and spinal cord. And you give it by spinal tap 3 times per year. And it goes in and homes in on one spot in the genome, and it changes the assembly of a critical gene called SMN2 that happens to rescue the effects of children who have mutations that cause spinal muscular atrophy. And this has been a life-saving and field-changing drug, showing that you can change the course of this disease with repeated doses of these by spinal tap. This is the second drug that I showed on the introduction slide, the drug targeting a gene called CLN7. This is a drug that my team made. This drug we made in 2017 and began treating a patient with in 2018, and it was a mutation-specific N of 1 medicine. What does that mean? This was a mutation-specific ASO that targeted a mutation we found in one patient. Her name was Mila, and she had a devastating fatal genetic disorder that caused degeneration of the brain. And this drug was tailored to that mutation, and it was made to treat her to show whether or not this condition could be treatable. And this project is something I've talked about quite a lot. It really inspired us to, it actually showed clinical efficacy and inspired us to try much more of this type of approach. And it has lit a lot of excitement around this idea of a programmable medicine and these so-called N of 1 trials. And N of 1 refers to the fact that these are hyper-precise drugs that target specific mutations. By the way, they're not always just for a single patient. These mutations sometimes are found more commonly in the population, so you might find it in one patient, you might find it in 10 patients, 100. Sometimes, if you're lucky, you might find it in thousands. But that's where the derivation of this name came from. And our motivation for running these trials is, number one, to help patients, many of whom are on that long tail of genetic disease, where companies aren't able to exert their influence. Our second motivation is that through these pilot efforts, we can generate pilot learnings for these diseases. Many of these are diseases that no one has ever treated before. And so developing ideas of whether treatments can be effective, whether you can get a response, whether the window for treatment is still open, really helps pave the way and for de-risking further drug development for other groups. And we also have other motivations for doing this type of work. One is we can improve our use of these drugs, get better and better at making ASOs, improve the platforms for making these drugs. And we can also begin to explore healthcare changes, delivery models for delivering this new type of medicine. I won't have time to go into that. Through count just a couple weeks ago, there have been over 30 patients now under this sort of N of one paradigm that have been receiving experimental medicines in this manner since we made the first one in 2017. And this has been the work of an incredibly large community, including many of the people shown on this slide. I had the good fortune of connecting to your community in 2018, just a few months after we started treating our first N of one patient. And Brad Margus at the AT Children's Project introduced me to a baby girl from your community who had had an abnormal newborn screen. She had a low Treg count, which is a newborn screen that is picking up many kids in the community and flagging them as maybe having an immune system problem. But like many children in your community, she did not have that immune system problem as the primary problem. She actually had AT. And this workup was done at Stanford and University of California at San Francisco. And we heard about this again through the AT Children's Project. What we noted was exceptional about this case was, in addition to her being picked up at birth, one of her mutations had an unusual effect. It created what's called a new splice site. What does that mean? In the middle of one of the coding blocks that contain the information for the ATM gene, block number 53, there was a sequence, a single letter change, a C to a T change, that introduced something called a new splice site. What is a splice site? A splice site is a little piece of code in the DNA that tells you that this is the beginning of a new block of genetic information, or this is the end of a new block of genetic information. And it tells you, like you're reading an article in a magazine and you reach the end of the page and it says, flip to page 53. Okay, flip a couple pages, here's 53, and here's more of the article continuing on. Well, this signal here, this new splice site, created a signal to the reader to skip ahead, but that signal came too early. It came within this block, this exon 53, 100 letters too soon. And it led to one skipping ahead and skipping an important part of the story. And this ended up breaking this little girl's gene and causing her to have Ataxia-Telangiectasia. Well, how did we use an ASO to try to fix this? This opportunity, we realized, could be taken advantage of if we could make an ASO that bound to this particular spot in the genome to hide that signal, to hide that skip ahead signal, to hide that new splice site. Essentially, you can think of these ASOs as molecular band-aids. They're coming in and masking this site and keeping it from being read by the genome. And therefore, the cell, when it encounters the mutation, instead of skipping ahead, it will ignore it. And it will be able to get to the natural end of this block of information and then continue on. So we designed a series of these long horizontal rectangles in gray that covered this region. And we designed a series of these band-aids, and we just tried a whole series of them, maybe 20 or so, and laid them out over the mutation. And the ones that were dark, the dark rectangles, were the ones that worked best. How did we show that they worked? Well, we don't have a mouse model of this child's mutation. So we used skin cells from our patient, and we grew them in the lab, and we tested the RNA patterns of the ATM gene in our hands. And what we were able to do is develop a way of visualizing the pattern of gene splicing. And we saw two different patterns that give two different bands on the gel. And these white bands that you're seeing, one is higher than the other, represent the normal and the abnormally spliced forms of the gene. And what I think you can generally tell is that there's a progression from the left to the right as we increase the amount of this drug, where you see more and more of the normal splice pattern of this gene. As we added the drug, the normal pattern of gene expression reemerged. We used data like this, plus functional data, where we took these cells and we stained them green to indicate when the ATM gene was functioning properly, to convince ourselves that restoration of the splicing pattern of this gene not only changed the splicing pattern, but also restored function of the ATM gene. When you take cells like this and you irradiate them with X-rays, it breaks the DNA. and it makes them glow green in the normal situation. But patient cells can't respond in this way and they don't glow green. Adding the ASO restores that green glow. And using data like this, we were able to convince ourselves, ethicists at our hospital, as well as the FDA, to give us permission to begin trying an experimental treatment for this patient's mutation. Our trial was launched at age 2 years and 10 months, so just a little shy of 3 years, after about 18 months of hard work with the team shown here. And before I describe the results of the trial, I have to take a short divergence. These are precision mutation-specific treatments. They're caused by mutations, in this case, that create a new splice site. And a very reasonable question is, how many patients like this are there? And how do we systematically identify them? Well, we studied this using a registry at the Broad Institute that have been created by the AT Children's Project. And the punchline is that we found that 15% of the families who enrolled their children in this registry were treatable using this kind of splice modulating, splice switching ASO approach. And one point that I'll make is that these patients had a variety of different mutations, and some of the mutations were more common in the population than others. In total, we found 35 individuals who were treatable using this approach out of 235. And of those 35 that were treatable, 13 of them had the exact same mutation. This is the one on the far right of the slide. This code C5763-1050A to G, it turns out it's the cause of 18% of AT cases in the United Kingdom. And there were other mutations that we found that had recurrence and were common in the population. And then there were a whole bunch of mutations that were rare in the population. By the way, our initial patient, the newborn that we designed that first drug for, her variant only showed up one time in this population. However, that's it's misleading. It isn't actually an N of 1. It turns out that her mutation is found in more individuals. It's just that our sample size was too small to capture it. Doing a little bit of math, if you look at the 35 individuals who have treatable variants in this way, you could design one ASO targeting the most popular mutation, and that would treat a third of the cohort. If you design several more, like 5, you could treat over 2/3 of the cohort. However, a sobering fact is that if you want to treat actually all of those 35 individuals that we found, you would have to make 15 of these different drugs. And so that's where the fact that these drugs are quick and easy and relatively inexpensive to make becomes really important. We have to find ways of using our scientific and regulatory know-how to enable making many of these drugs at once instead of one at a time. Okay, back to our trial. Our trial was initiated just a little bit before age 3, and Claudio de Guzma was our lead principal investigator for clinically overseeing this trial. We had a complicated clinical trial plan. We began dosing in the red on this graph here. You can see our doses. There's a dose escalation scheme where we began with very, very small doses to be safe, and then we gradually escalated that. Doses were given approximately every 2 weeks in the beginning, and then we extended to every month, and then we extended to every 3 months. And as you can see, these bars go up and down as we've changed the doses over the years, as we make minor adjustments to our plan. So we've done this now dosing 3 to 4 months at a time, every 3 to 4 months at a time. And we started in someone who was just about to turn 3. But as folks may know from this graph by Dr. Tom Crawford, typical severe progression in AT begins around age 5. So we knew that we would have to be doing this for a while to watch and wait to see if there were long-term effects of this condition. Our trial began here at just before age 3, and it's been running for 5 years, and our patient is now at age 8. And the punchline for what we've seen so far is that 5 years later now, for this child at age 8, we have seen really remarkable stability of her neurologic symptoms. We've seen no real signs of neurologic clinical progression. 5 years in, we've been following her using various clinical rating scores, like this one, again made by Tom, the AT-NEST score. And she's been outperforming age-matched children with classic AT. These graphs here show the distribution in gray dots of age-matched children. Our patient here is shown in the red star, and she's doing at or above her cohort level in all of these domains. We are also following things like wrist and ankle movement sensors. And these are complicated sounding but simple looking devices where you wear a watch on your ankle or your wrist, and you use it to track movement patterns. And this is the brainchild of Anupam Gupta at MGH, a talented neurologist there. And you can use the movement patterns to separate neurotypical kids from AT kids. Gray dots, red dots represent what the normal patterns of these movements are over a range of ages. Our patient is performing in the blue dots right here. She's in this upper end of the AT scale, overlapping with the neurotypical scale. And interestingly, when we look closely at this graph and we say, well, of those AT patients, how many of them might have mild mutations that make them milder than classic AT? Those mild patients live right around there. So our patient's performance is clustering with the mild mutations as opposed to the classic patients. We've also done other things like brain imaging to see if we can see that we're slowing the cerebellar shrinkage. And the punchline there is that, well, we're not sure. It looks like it might be slowed, but when we use quantitative automated computer algorithms to calculate the volumes, we can't necessarily tell the difference. But we're looking hard at this with Rob Dineen, Ellen Grant, and others to see whether or not this might be an artifact of the computational algorithm. Because to our eye, it looks like it might be improving, but we're really not sure. We've also been working with the Johns Hopkins Clinical Center for AT and talented researchers like Valerie, Natalie, and we're seeing some unexpected systemic effects. So this is a treatment given to the brain and spinal cord. But really interestingly, Val has shown that children with AT don't grow at the same weight, at the same rate, and they don't gain weight at the same rate. That's hard to say, as the neurotypical children. And our patient began in that same way, very, very small for age, but with treatment, she's actually shot up several percentiles on her growth charts in a way that was actually rather surprising to us. We've also seen some biomarker changes too that are surprising to us. AFP levels in the blood normally rise in AT, but in our patient, they aren't rising. They're refusing to rise. Well, they're going up a little bit, but they're again crossing several percentile lines. And this is really an intriguing effect that we don't fully understand. So let me wrap up. I'm sorry for going a little bit over, but let me say some important limitations. This remains experimental. This is not an approved drug. We only have long-term results from one patient so far. We have additional results from a second patient who's being treated. And there have been some side effects, like increased CSF pressure, although thankfully without any symptoms. In addition, this specific ASO only works for children with this specific mutation. And we found a dozen of them in the world, but a dozen is a start, but it's a drop in the bucket. Overall, 15% of patients might someday be treatable with this strategy, but we need to make new ASOs for them. Still, despite those caveats, here are the takeaways. And precise mutation-specific ASO treatment, we think, may actually be effective for AT. And that really gives us a lot of motivation. It motivates us to expand this approach. It motivates us to enroll more patients with this specific mutation. And it motivates us and a growing group of people to make more ASO drugs for more eligible mutations, including, for instance, this mutation which is so common in the UK. Most broadly, it suggests that AT can be treatable after birth with good effects. And that motivates us and others to keep trying both not only this approach, but other approaches too, and gives people a push to say that this could be possible based on these pilot results. So we hope that this is a useful contribution to the field. And again, sorry for running a little bit over, but there are many people to thank, including most importantly, the family of our patient who's demonstrated incredible trust in us as we take this responsibility really seriously. Thanks so much.